Hey everybody, welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me from New York, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. I would ask you how you were today, but I saw you for a while.
I saw you from like 11:30 till 5-ish, right?
Yeah, but there was like a 3-hour in between period where, you went to a defense, I wrote a grant, you went, did some stuff, I did some stuff and wrote a grant, and then I complained.
Let me
Which
give,
is basically my day.
it's all right. Amy
Every
likes to
day.
do that. Amy likes to do that. A couple of updates. First of all, how cool is it that we have just passed 99,000 subscriptions on YouTube?
It's
Wow.
great.
You know, there
So
was a
maybe by
time
the weekend we could be at 100.
before COVID, I couldn't get past 20,000. And it's COVID that brought us up here, and also you guys on this stream. And I appreciate it. Lex Friedman podcast helped. That gave us quite a bump. Anyway, probably by the weekend we'll bump over. We've been getting about 100 a day. It's cool.
It's great.
Virology for the world. And the other thing I wanted to show is a little bit of progress on the incubator. So this is where we are right now. I am actually able to record, and tomorrow I was hoping to record the first episode from the incubator for Daniel's show, because that he has actually made this possible to rent this, to lease this. I'm not sure if that'll happen though, but we'll see. Anyway, everything you see, folks, the equipment, everything but my jacket. Okay, the equipment, the camera, the monitors. This is only part of what's going to happen. The sound panels, all of that is bought with listener contributions. I could not do any of this without your contributions. And of course, once we get it set up, then that kind of spending will stop. But this is great. This is the future of, or a future of science communication. And by the way, the colors picked by Amy.
Yeah, we're going to go hang them on Saturday after I go to the Apple store and yell at them for not fixing my stuff.
What didn't they fix?
I don't want to talk about it.
Okay. All right. Now, folks, we're going to dive into your questions. And the way I do this, some of the earliest questions disappear. And they're all good. So if I don't get to them, like the first one are people. So I need some mods for next week, because Virology Live is beginning next week. And Vanity Nutrition has already volunteered. Thank you very much. What does it take to be a mod? You have to be able to spot people that are, how should we say, not really interested in being here. They just want to disrupt. All right?
Well, they're disruptors, but I think you should tell them what time you're doing it so that they can decide whether or not that's an appropriate time in their schedule. Time that you're going to do this Virology Live and how long. It's a commitment that they have to make. So,
For example, there are already 2 people here that have given us a thumbs down, and we haven't done anything yet. And those are the people you need to look out for, because they're just being disruptive. And I'm learning slowly not to pay attention to them, but they can, interrupt a nice flow. Anyway, the Virology Live will happen on Mondays and Wednesdays. The first class will be Wednesday next week. It's going to be 11 a.m. Eastern Time, U.S. Eastern Time. All right?
and how long?
2 hours maximum. So what
Okay.
we're going to do is I'm going to teach you virology. I am going to pause periodically and give you a little online quiz, right, to see how you're absorbing. And at that point, we'll take some questions relevant to the lecture, okay? As I said the other day, if it's a lecture on what is a virus, please don't ask about vaccines. Yeah, save that for the vaccine lecture. That'll be a challenge, but that's fine. And after,
Okay. So
I'm
now
going to put
they can make.
I'm
Go
going to
ahead.
put up a syllabus, make a web page by the weekend, and then you will have exams. There will be at least a midterm and a final, and they will be graded to see how you do in virology. And, maybe I'll give out prizes for good grades. Who knows? Anyway, so I need a couple of moderators. And,
well, don't forget that we have 2 grants due in October.
Don't worry, we'll get them out. Would you rather I didn't teach the course?
No, I'm just saying you have to organize appropriately. Now people know what time it is, how long it's going to be there. They can make an appropriate decision about whether or not they can modulate. And we need to moderate whatever. And we need to reorganize our time to make the 20-page deadline on the 10th and then the one on the 16th. Otherwise, I will be very unhappy. And we don't like that. When I'm unhappy, it never works out well.
Anyway, there are 2 mods on this live stream, Vanity Nutrition and Frank P. And you can always ask them how they like it and what they have to do and so forth. But if you have any questions, just shoot me an email, vincent@microbe.tv. I don't know. You can't have too many mods, right?
Apparently not if you turn on the thing and you get told you are a thumbs down. Although I think, people just do thumbs down just to be contrary.
That's fine. Oh, name your top 3 trusted scientists in the SARS-CoV-2 arena. Amy Rosenfeld, and Amy Rosenfeld.
No, I'm not really a corona person.
No, you're not.
No. Although
why don't you name your 3 and I'll name my 3 because I suspect they'll be different. Do you have 3?
No.
You don't trust anyone?
Well, I'm not really clear how to interpret the word trust. I have a couple of people that I think have done good work. And interesting work that has either led to reagents that are critical to understanding virus biology or disease.
Mm-hmm.
And one of them you will be surprised at because I immensely dislike.
Well, now you can't tell us who it is.
Yeah, I know. Can't tell you.
You're not going to name anyone?
So I think so it's really too heavily focused on spike and antibodies and stuff. But I think like the conglomerate of SETI and CROTI have a good handle on the whole immunology picture, B cells and T cells.
Mm-hmm.
So
I agree.
I would say that they are not a separate, they're one entity that I would trust for, that I think has done really relevant work.
Okay, I agree with that.
I think some of the interesting beginning animal models that came out of Rocky Mountain were critical and helpful.
Yeah. So that would be like Vincent Munster and Emma DeWitt.
DeWitt and Heinz Feldman, and all that whole group of people.
Okay, I agree with that.
And then I think that there's a group of people who made who haven't really, I think that there's another group that hasn't really published a lot, but he is definitely an expert in the field. I think Stanley Perlman has a lot to say. But actually I think I would put Susan above him. I
was going to say Susan, yeah.
Yeah, I would put Susan way above him.
I agree.
And it's, for me, it's irrelevant of whether or not you have a paper every other month or not. It's really the quality of the work. And I just think that she's awesome.
Okay.
I think she's really amazing.
Good. On journal papers, could you decode the significance of first authors and last authors, please? Dr. Rosenfeld.
Well, first authors are the people who are supposed to have done the majority of work and whose project it was, and all the other people are supposed to be accessory. And then the last author is the senior author, and it's his lab that actually the work was carried out in.
And sometimes you can have multiple of both, right?
Well, nowadays you have multiple of both, but it's never really honest to have multiple of both, because one person always does more work than the other.
Okay. Not much decoding needed. Thoughts on Pfizer doses for young people. 11-year-old gets 10 micrograms, 12-year-old 30. What do you think, Amy? That's what they tested, right?
Yeah, it's what they tested. And there must be something about that transition between 11 and 12 that the majority of children, that's either the beginning or, a really large growth spurt occurs in the year of 11 to 12 that allows for that. I mean, there's got to be a lot of physiological changes at that point. That's the transition. So I'm sure it wasn't a willy-nilly decision.
As a teacher, how do you think you could bridge the political divide between science-based debate and dogmatic rhetoric going forward?
What is dogmatic rhetoric? Is that like there's the fundamental principle of biology? You go from DNA to RNA to protein?
Well, if you're dogmatic, you only have one view, right?
Well, there is only one view about that. It always goes from DNA to RNA to protein. Now, you don't have to start at the beginning, but you never go from protein to RNA to DNA.
Yeah, I'm not quite sure. I mean, I see on the one hand science-based debate, but what's on the other side? I don't understand dogma. That means people who are not willing to listen. It depends what you're talking about there. I mean, if you're thinking we're not listening to the lab origin notion, then no, because that's not science. But maybe you're thinking of something else.
Well, I think the paper, are we discussing the paper on Friday?
Which paper?
The one about the bats in Laos.
I don't know. You have to pick. Tomorrow I'll pick them. We can discuss.
But that paper clearly demonstrates that it doesn't come from a lab because she didn't sample Laos.
Well, it's only the RBD, right? That's highly similar.
No, I think it's the whole genome.
All right, we'll see. If that's the case, it's important to do. I agree.
Right, but the other thing is, even if it's the RBD, she wasn't in Laos.
No, I know that. I understand.
So right then and there's no need to debate the lab origin hypothesis. There's no need to have a lab origin hypothesis. It's debunked right there. Yeah. It's the end of discussion.
Thank you for your super chat, Mr. Ozzy Cam. I'd be happy to volunteer. So all of you who are interested, Vincent at microbe.tv, and we'll see who's serious. Okay? Because I think both with Vanity and Frank, I communicated by email, and I talked about what my expectations were. I don't, if there's more than 2 or 3, we don't all need to be there. Some of you might not be able to be there every time, but let's have a discussion offline. Assuming 25% of us never get vaccinated, how long do you think it will take for SARS-CoV-2 virus to become just another cold bug? Amy will not answer this.
No.
She doesn't like to predict.
No.
So, Ralph Barrick, back in February, he said, I have no idea. Could be 50 years, could be 100 years, it could be 200. He has no idea. Really, no idea. Sorry. Okay, Jim Frank, I got 2 doses of Moderna in February and March, 76. Today I started on immune therapy, Keytruda.
Do you know what it does?
It's a checkpoint inhibitor, right?
I don't know. That's what I'm asking.
Yeah, Keytruda is a checkpoint inhibitor, I believe.
So then why is it immune therapy?
And it's for melanoma, lung cancer, head and neck cancer.
Yeah, but why would it be considered immune therapy? It's not an antibody. It's not CAR T cells. It sounds like it's a chemical.
No, it's, let's see what it is. A humanized antibody. Yeah, it's a humanized antibody. Is there any reason you started immune therapy? Should I take a third shot?
Go ahead.
I don't think it would hurt if you feel better. Right? And by the way, if you want to direct your questions, put an at VNA. At VNA is fine. Just so we know it's that as opposed to you guys chatting amongst yourselves. How long have I known Amy? As she said the other day, are you tired of knowing me? Amy was my PhD student, I don't know, in the 90s. What year did you come to the lab?
99. Well, you gave a lecture in a class I took as a first-year graduate student.
Mm-hmm.
And in February of 1999. And then I came to do a rotation in May.
And then when did you leave?
I defended in 2005. I went to Montreal.
She went off for a number of years, then came back to the lab in 2015. And that's the second incarnation of Amy.
Yes, the second incarnation of Amy. But it's ironic that I've known about you for more years because I know Bob and Eric before I knew you.
Yeah.
Plus, I knew Peter before I knew you. And Jim.
So there are all kinds of connections. So Amy was a tech in a lab in the same department where I got my PhD.
Yep.
And she knew people from my lab who had gone to that department. So it's very interesting. It's a tangled web of viruses or something like that.
Yep.
All right. For transparency, shouldn't Daniel report on vaccine risk frequency just like COVID? Well, he doesn't give the numbers, but he says, the risk of myocarditis is far, far lower with the vaccine than with COVID. You want him to give the numbers? I mean, I'll tell him. He might do it, but he might not. Let's see. Amy, Vincent, this week's theme is boosters don't work in Israel. What do you think, Amy?
Was there a paper that said that?
It's a press release or something like that, right?
Oh, really?
Yeah, I saw it mucking about on the news.
I don't know.
Now, listen, you have to take everything with a grain of salt. Observational studies are hard, as we talked about on Twiv a couple of weeks ago.
Actually, they're easy to do. They're just hard to understand and come up with the right interpretation of the data. But that's a separate issue. We could do a lot of observational studies. I mean,
hello, Frank.
Easy.
Thanks for being back, Frank. How does the vaccine for lung cancer from Cuba work? I have no idea.
There was a
I didn't know there was
vaccine.
one. I'm sorry. How many TWIVs there's this week in virology, this week in microbiology, parasitism, evolution, neuroscience, and immune. Immune is the only one without a this weekend. I wanted a one-word podcast. I wanted to call it Node.
Nobody would get that.
As in lymph node, right?
Yeah, I know what it is in reference to, but I don't think people would have gotten it.
But yeah, it's immune. I like immune. It's good. It's a good one.
And there had been urban ag and various other things.
Oh, yeah. We used to have one called urban agriculture, which was Dixon's idea, and then he didn't nurture it. So it got to 29 episodes, and I said, that's it. I'm not getting guests. This is not my field. It's your field. And he said, I can't get guests. They don't agree to come on. I said, well, I have no problem getting guests on my other shows, so I ended it. On a recent live stream, you were asked about beneficial viruses, mentioned polyoma and circoviruses. On Wikipedia, they describe polyomavirus as oncogenic. Well, in the wrong host, polyomaviruses are oncogenic, yes. So SV40, a simian virus, is oncogenic in other species. But the polyomaviruses that we have that are coursing through all of us are not oncogenic in us. Maybe they would be in another host. But they're with us and they don't do any harm. That's why I speculate that they're beneficial, but we don't really know. Okay, this is a tough one. Would you please comment a little more in depth on those presenting like credible sources like Brett Weinstein, Stephen Key, etc.? Scientific reasons for the likelihood this is a lab leak. So these are people who don't know any virology. Okay, and I don't mean to be offensive, but Brett Weinstein is an evolutionary biologist. He doesn't study viruses.
Does evolution biology of like limb development, which is totally different. But you don't even need to be a virologist. You just need to have some common sense.
I mean, there's no scientific evidence for the lab notion. On the other hand, evidence is building that this is a natural virus from animals in nature, and we're seeing more and more where it might have come from. There's a paper this week Amy was mentioning earlier where highly related viruses to SARS-CoV-2 have been found in Laos. And,
which, if I believe correctly, is south
of China. So, on the other hand, in my opinion, the lab leak notion is just built on politics and hearsay and no credible evidence. So, in fact, Weinstein and Key have no evidence for it, and I don't understand why they're doing it. Maybe they like the attention. Weinstein has way more followers than I do, so there you go. I mean, not that I'm complaining, but I don't understand it.
Well, because the parking lot got full. So it's like, so there must be some horrific event at the Short Hills Mall in New Jersey between November and the middle of January because the parking lots get full.
Yeah.
Shocking.
So let's see here. There's another one here. Carlos, I have a friend just finishing nursing. They're worried about working directly with COVID patients at bedside. How risky is it for them fully vaxxed to bring home to an unvaxxed child? Well, this is the main worry of a lot of people who are facing the public, like even professors now who are teaching in front of classes. Even though they're vaccinated and masked, they're worried they're going to bring virus home to their young ones.
Or their older ones.
Or their older ones, right? To which I would say you have to be very careful. So you're vaccinated, which is good. Make sure you wear your mask around people all the time. And when you go home, make sure you wash your hands before you touch anyone. And if you want to keep your mask on at home, that's fine, but I would change it. Right? Go somewhere away from people and change the mask to a clean one that you just keep at home. What do you think about that, Amy?
I think it's good advice. I'm just not sure what they've been doing when they've been going outside, to the grocery store or whatever. And because those people in the hospital don't have any virus.
Yes.
The virus has already cleared. Those people who are in the grocery store who are potentially asymptomatic and not vaccinated have virus.
Yeah, so it's not just
so I'm not clear, right? So I'm not clear what, people, I just, I'm not clear.
This time next week, you have likely cracked 100K subs. Probably, I'm sure we will by next week. I think you missed your chance to get through all the questions. Go on Joe Rogan and see how far your reach can expand. Do you think we could reach 1 million, Amy?
Tomorrow?
No. I'm willing to wait 10 years.
Oh, yeah, sure. Yeah. No problem. Tomorrow, maybe not.
No, we're at 99,100 right now. So it's fine. Listen, I just think it's exciting. 100K is a good number, and for science, and the kind of science we do, right? It's hard. And to have that many people, it's really good. I like it.
Yeah.
By the way, Amy's going to be on TWIV Friday for all you Amy fans.
Yes.
Is that right? Right? You're going to be on Twiv, correct?
Yeah. Neither Kathy nor Brienne, so I'm the default female. Yeah.
No, you're not
No
the
problem.
default female.
It's
You're a voice
okay.
that we
It's
tap
okay.
from
It's
time to time.
okay. It's okay. I don't mind.
I never call you default. That's nothing.
It's fine. I'm not clear what a saline nasal rinse would do.
It's only temporary protection, yeah. It would rinse out what's there, but it would immediately... So you're talking about after exposure. How do you know you're exposed to someone with COVID? It seems to me you'd have to be rinsing your nose out constantly, depending on where you were. So, and I think, if the virus has gone deep, you're not going to get back there. So I don't think it's useful. On Face the Nation, Scott Gottlieb warns against publishing the sequence of future novel viruses because it provides the recipe to a rogue actor to make that virus. Well, the virus...
Really? wait a minute. We've been publishing sequences since the beginning of time. Your postdoc project has been in Genping for 40 years, at least.
That's right. That's right.
When was the last time you saw somebody make a virus in their garage? Because let me tell you, if you can, I want to hire you because we're having trouble.
Yeah, rogue scientists.
And we have all the technical equipment, and we're very good. I'm very good
this
at
it. This is where some kind of experience helps, because we know it's really hard to do this. Even in our hands, it doesn't always work. So a rogue scientist wouldn't be able to do it. And anyway, what would they do? You want to get SARS-CoV-2, you just find someone who's infected and do a nasal swab.
This is the stupidest idea ever. Why don't you just inhibit all research whatsoever? Why don't you just say, we don't need to have research whatsoever. We should do none of this. We should close down the entire research enterprise. And good luck to you when you get a new disease or you need a new drug. Let me know how that works out for you.
And here's another one. With so many scientists, should we be worried about weaponizing? No. Nobody has ever been able to weaponize a virus. It's too hard, and I'm not worried about it.
Really? Like, this is what people think about? So many scientists now working on COVID? Really? This is what we, like, oh my God.
I have not had a chance.
I have a question. There's so many business people in the world who are working with money. Should we worry about the fact that we're all going to go broke? My bank account is going to be drained by somebody who pushes a button mistakenly? That has a higher probability. I believe some of the crashes that we've occurred in the stock market in the early 2000s were because somebody pushed a button, or maybe not a crash, but like where they've had to stop trading in the middle of the day was because somebody pushed the wrong button. Yes. No, I'm serious.
I know you are. I know. It makes the virus questions look so remote, right?
Yeah, that's my point, right?
Yeah. No vanity. I haven't heard from Tim. I should email too, but you have to jog me, okay? So you shoot me an email and remind me.
What is Tim?
A cappella science. These are these people who sing a cappella science songs. We'd love to get them on Twiv and do a song. That would be cool.
What's wrong with the opera people?
There's nothing wrong with the opera people. We want to just get more people, that's all. Does kinetic of infection infect spreading from the vaxxed? Would T cells clamp down on shedding after 2 days and quickly limit the ability to transmit? Well, from the Singapore study, it's very clear that vaccinated people who are infected, their RNA levels determined by PCR from nasal swabs decline much quicker than RNA levels from unvaccinated people. I don't know what's doing that, right? It must be a memory response. And so it's probably a combination of antibodies and T cells, wouldn't you say, Amy?
Yes.
A little love for Amy. Look at that.
It's cute.
Isn't that cute? Wouldn't a vax with spike and additional nucleocapsid help with broadening the T cell response? Absolutely. I've heard of industry people talk about that. Next generation vaccines. Brand new vaccines. COVID-19 deaths in China, 4,700, U.S. 670,000. Why? Amy, why?
Most likely because of lockdown, and they have a serious following policy. Like, they have a serious quarantine policy, masking policy, lockdown policy. None of this stuff, and they wait until it's to a degree, I think, like several days, several weeks before they release people from these lockdowns. They don't just say, oh, well, we've been in lockdown for 3 months and it's destroying our economy, so we should just all run around wearing no masks.
When they say lockdown in China, you have no choice. They check you. You check where you go on your phone. It's a kind of surveillance we don't take here, but that's one reason.
They do that in Spain too.
Oh, yeah? But the other thing is they clamp down on travel. So if you go to China, I don't know if this is still in place, but in the past, you have to quarantine in a hotel for 14 days. And you have to pay for it yourself, and you have to pay for your meals. I know because I know scientists from China who went back and forth, and they said, yeah, it's a problem, but you have no choice. He said they bring the meal on a tray, they put it outside your door, and you pull it in.
That's what they do when Canada, before Canada opened the border, was you went anywhere and you had to, But you also didn't go anywhere, even within the country. There was no, during that period of time. Like, you didn't go from, like my sister last year came for 7 weeks from San Francisco. So in May 2020, she came for 7 weeks and then she went back. There's no going from Beijing to Shanghai like that in China. That's just, but it's totally different. And I'm not even sure, to be honest, that 7,000 is really the right number.
Yeah, that's the other problem. Yeah,
the right number.
I agree.
But I don't know what the right number is. But like in Spain, my haircutter's sister lives in Spain. Her neighbors would tell on you if you went out more than the hour a day. No, I'm serious.
I believe it. No, I have not gotten a third dose. I have no plans to get a third dose, even though I'm eligible or not? Did the FDA approve
you're
over
65?
No, I'm not. I don't. As I've said, I do not think a booster is needed. So I'm not getting it. Do you think people with natural infection have the same clearance level of vaccinated people regarding vaccine passes? Big discussion here in Brazil. My understanding is that the immunity after recovery from infection is more heterogeneous than after vaccination, which means Franz, no, what's his name? Kramer? No, Kramer. What's his first name? Florence Kramer. I did a TWIV with him last week, which is being held back, but it should be released soon. He said, some people make a great response, some intermediate, and some none. And we don't know which of those you would be. So that's the argument for getting one vaccine makes you the best immune person on the planet.
Right, you don't know which one you would be even when you get a vaccine.
That's true.
So the argument doesn't make any sense, actually.
Well, maybe if you had no response after a boost, you would have a really good response. You don't know that, right?
Well, first of all, you don't know if you're going to respond to the virus if you naturally get it.
All right, but anyway,
Number one, you don't know that you're going to respond to the... Number two, you don't know if you're going to respond to the vaccine if you get the vaccine. And number three, if you didn't respond to virus, what makes me think you're going to respond to the vaccine? So I don't understand the argument. The argument is flawed.
Are you saying that you want people who have recovered to get a vaccine pass?
I see no reason why, if you have recovered, you are not considered equal to somebody who is vaccinated.
Yeah, that's fine. I have no disagreement. I just think I was just presenting what the argument for.
Right, but I just don't think it's well thought out. I don't think it's completely well thought out because it doesn't make any sense.
Amy looks more rested. Actually, both look more rested. Amy, are you more rested?
Sure, we can go with that. Was that convincing?
No. When Amy says sure, it means she doesn't agree. Okay? I know that from experience. When will vaccines get the same? Yeah, it's the same question. I think.
No, he wants to know. No, it's not the same question. So he wants to know if you like. So if you if you have X amount of protection after COVID, and the vaccine is not as good, will you develop it, or you need like 3 boosts in order to get to that?
Okay.
That's the question. And nobody really knows because we don't do those kind of studies. Or at least haven't followed those kind of studies.
Anyway, thank you, Ozzy Camp, for your contribution. Shoot an email, Vincent@microbe.tv. We'll chat. I always wondered what happened to the virus when the host is dead. Did they have to incinerate all the infected pigs, or can the meat still be eaten when it is cooked? Well, no, they usually... So in the case of Nipah, which had an outbreak in Malaysia, and they culled 1 million pigs, they just buried them. They didn't burn them. They dug a huge pit and they pushed the pigs in, and very sad, they just put dirt on top of them and smothered them all. But you can't eat them, no, because just in the process of preparing the meat, you would contaminate yourself, right?
Well, I believe that they do the same thing with avian flu here, is they cull all the chickens.
That's right. You had to get J&J recently after reaction to first Pfizer. How protected am I? Will I get T cells?
You're very well protected. Remember, you and I did that Nature paper.
Yeah.
So they have T cells, they have B cells. They're very well protected. Remember that Nature paper?
Yeah, J&J is now exploring a second dose of J&J.
Right, J&J but vaccine, it's not good if you had some kind of allergic reaction.
Nope.
And there's a big range for what an allergic reaction is, so I don't really know.
Is someone more likely to catch the common cold or COVID this fall, winter? I know Amy's thought about this. What do you think?
You have equal probability, is my prediction. Masks reduce the amount of cold transmission and COVID transmission. So once you get rid of masks, you have the same probability. And especially since there are so many more common colds. Like there's 160 different genotypes of rhinovirus. There's one genotype of SARS-CoV-2. And I learned from Anne the other day. I thought that this was really interesting.
And who?
Palmenberg.
You have to tell these individuals here don't know.
No, Palmenberg. She gave a talk in the meeting that I and the chair of micro at North Carolina host together. And so she said that when you take a nasal pharyngeal swab of any healthy person, you automatically find Rhino B in them, because it's a benign infection. So automatically we're all infected with Rhino Bs. So right then and there, you're more likely to catch the common cold because we already are continuously infected with Rhino Bs.
I think this winter many people will feel common cold-like symptoms, and some of them will have rhinoviruses and other common cold viruses, and many of them will have SARS-CoV-2.
I think fewer of them will have SARS-CoV-2.
Perhaps because we're actively vaccinating, perhaps, Amy?
Yes.
Could SARS-CoV-2 become our go-to winter virus, replacing flu?
No.
No. I agree. I think we're going to have them both. Oh, by the way, people love the colors.
Oh, it's good.
There you go. Love the red color. And we have some more sound panels coming in that are in the shape of a virus. And Amy picked those colors too. Those will be cool. Let me show you what's going to happen here. So this area right here behind the camera, kind of next to
a little pointer person. Do you have a pointer person?
No, I don't. Sorry, because it doesn't work here. I'll just say that behind the camera, you see there's a gray, red, red, gray, red. That wall is going to have 2 interview chairs and a little table between them. And behind that, on the wall right adjacent to the window, we're going to put an arrangement of sound panels that are in the shape of polygons, which look like viruses, right? So that'll be, when people come to visit and I say, hey, let's do an interview, they'll sit there and have the polygons behind them.
I picked the furniture too.
And Amy picked the furniture. We went out to what is it? Design Furniture Warehouse. What's the name of that?
DWR.
Design Within Reach. Anyway, it's going to be cool.
Yeah, it's very mod. A lot of what did we get? A lot of Knoll and a lot of Ames and some Noguchi.
Yes. Very nice furniture. When it's all there, which should be in the next few weeks, you'll all see it. All right. Vaccine efficacy for Pfizer, 91%, declined to 77. Thoughts. The thoughts are that's all COVID. If you look at severe disease and death, it's still in the 90s. All right. So you have to make a specification of what you're talking about when you talk about VE. Right, Amy?
Yep.
I pity the Apple Store. Why is that? I don't get it.
Because they're concerned that I'm going to yell at them since they didn't fix my
that's right.
First time. They don't yell at people.
No, in public she keeps it good. Where is this going? Amy goes till about 10 minutes from now. And, oh, no, you're not asking about that. I know. I'm just joking. Where does it go? How does it end? Eventually, most people will be either infected or vaccinated. And I think it will be a winter disease, but I'm sure Amy will disagree. I think we'll have outbreaks every winter among the unvaccinated, far less extensive, and we'll just live with it.
I don't disagree that it's going to be a winter disease. I think most respiratory diseases, except for the one that we work on, are winter diseases. Ours is just unique, but it's unique in many different directions, right? So you can't really go off of ours, but the majority of them are winter diseases.
Wow, I got a lot of volunteers for modding. Thank you, folks. You're really kind. I appreciate it. And all email me, Vincent at microbe.tv, except Frank and Vanity. Of course you don't, because you're already mods. So we'll automatically put you on the course. And everyone else email just so that we're on the same page. Does CT less than 24 and positive antigen test still represent infectious viral load? Or is there a new coefficient for each variant? As far as I know, no one has done the calibrations for the variants, right, Amy?
No, they have not.
So I would say assume it's the same. I am immunocompromised. Oh, look at the cat in the snow outfit. It's just adorable.
That's a cat or a dog? It looks like Gordy.
It looks like a cat.
Okay,
Aww.
It's a cat.
I've been advised to get a third shot. I got 2 doses in January. Is there any reason my third shot has to be Pfizer and not Mrna?
No.
You are exactly the right person to get a third shot. Okay? That's good. And either one would be fine for you. And Lucky Sixes. I'm good at spotting trolls. Good. You have a troll sensor. Good. What is Virology
Everybody
Live?
needs to have a troll dar.
Virology Live is going to be a virology course taught via live stream, right? I teach a virology course in the spring and summer at Columbia University. I am now going to teach it starting next week here on YouTube live stream. We're going to see how that works. We're going to have a few hundred people, and they can ask questions, and it just makes a broader reach for me, and it's live, which is different from watching a video, right? So that's what virology, I decided to call it that, and it got the Amy stamp of approval, Virology Live. So I have to make a logo. I guess it's going to be virology with like live crossing it.
I don't know. I kind of thought it should be like a bunch of viruses looking like that. What Sgt. Pepper cover of like where the Beatles are all walking along the street. Do you know what I'm
Yeah.
talking about?
Yeah, I
You
do.
guys did it for the back of the POV one year.
Yeah, I do. Katrine Wallace states the base rate fallacy is responsible for the atypical Israeli data. When adjusted for the base rate per 100,000, the rate of severe COVID is lower in vaxxed people. So I've heard this also from people who say that the numbers aren't being crunched properly. So we'll see. You know, doing things in a
Shocking.
rush is always a problem. Can you be a mod and learn? Of course. Just ask Vanity Nutrition and Frank P. They mod and learn. Here we go. Vaccine risks again. All right, let me see this. Never concern yourself with thumbs down. Every one of them is actually gamed by bots. There are no exceptions. Really? A bot is doing that? How? And they respond to me? Wow. Wow. Are you making Twiv polo shirts, or better yet, polio shirts? We have Twiv shirts, although maybe not polo. What is a polo? Is the one with the collar and the little
Yeah,
button
it's what you
placket,
wore
right?
on Saturday. It's what you wore on Saturday.
Okay. Yeah, we could make a Twiv. CafePress.com/Twiv. I will upload a polo shirt. Yep. Yep. Oh, so your live stream might be competing for search results space with someone totally unrelated, so they thumbs down it to improve their own rankings. Well, you guys are here because you want to learn, right? I am a 69-year-old, got second Pfizer shot, 3:30. How soon can I get the booster? Amy is the booster interval person.
So 9:30 would be 6 months.
Yeah.
So
Which
therefore,
is like, you can get
on
it now,
11:30, yeah, 11:30 would be 8 months, and they'd say you should get vaccinated in 8 months. You should get a booster in 8 months.
You have recommendations for books to read on Virology 101. Yes, I do. I will put them on the course webpage and let you know. I can't think of them offhand. If I do, I'm going to pause because I don't have any Virology 101 books on my shelf here.
What do you mean?
Shocking.
You don't have a POV?
It's not really 101.
Of course it's 101.
I think Lucien wants something a little bit.
Like Albert's?
No. Something like, there's a author by the name of Sompirac, okay? Let me bring it up here. Actually, this is a good recommendation. How viruses work. How viruses work. Sompirac. Yeah, this is it. You should check this out. I've heard this is very nice. Okay, How Pathogenic Viruses Work by Lauren Sompirac. Cheap, and it's really good. And it's 101. Totally 101, okay? You can try that. Okay. And what else do we have here? I get asked this by family all the time. If someone has end-stage heart disease and they get COVID and dies, should that really count towards the COVID death toll? What do you think, Amy?
Well, you don't know when they would have died from end-stage heart disease, right? So you can't really say that having SARS-CoV-2 infection didn't facilitate their death.
Yeah, I would agree. What is OG? I look like an idiot now. What's it? Do you know what an OG is?
All the OGs are volunteering to mod. No, I don't know. But
I'm looking at the
it's
chat here.
8:50. It's almost 8:50, and I have to play it out myself. So if we're gonna do a few more questions, let's do a few more questions.
All right. So now we have more on the Israeli data. It looks like Simpson's paradox explains the lack of vaccine efficacy. Could you have an epidemiologist on? OG is old gangster.
Old gangster.
Or original gangster. Yeah, we can get Jeff Shaman on to do that. How's that?
He has a paper in Nature Communications today, too.
Is it good?
It's Bayesian modeling. It's fine. If
I
you're
think
into Bayesian modeling, I'm not into Bayesian modeling.
I think Amy should be a feature profile for the New York Times Sunday edition's How I Spend My Sunday. Yes, I agree. There's probably no one who spends their Sunday like Amy, right?
Probably not. But
I mean,
I think the last one I read was Lady Gaga's mom. She goes, I walk past her restaurant all the time, and I walk past the Muffin Cafe, and then she walks the dog. It was like, okay. Oh, and then maybe she talks to the girls, her 2 daughters. Yeah, something like that. I was like, wow, this is just like my mom. She's just a mom.
all right. So you want to do a couple more?
Yeah, sure. And then I have to play it out myself.
Let me ask you one more question. Last week after you left, someone wanted to know what was the coolest thing you saw at the Met Gala.
The coolest thing I saw at the Met Gala?
Yeah.
I don't know that I would say that there was a lot of things that were not so good.
You liked Susan, is that Susan, the lady with the blonde hair and the black dress? You liked her, right?
I thought Sharon Stone looked fabulous.
Yeah.
But like Kim Kardashian came completely head sheathed in black by Balenciaga. I'm not really clear what she was saying. Debbie Harry came like
Yes.
in a hoop ball skirt of the American flag with not so good makeup. It was kind of scary.
Yeah, so you
Jennifer
like this,
Lopez,
right?
yeah, I thought she looked amazing. Jennifer Lopez came in like some cowboy uniform from Ralph Lauren. Yeah, if you're into the American West, sure, she would have looked. I guess you have to like that look. I mean, it wasn't my favorite look that she's ever worn. I didn't mind. Harris, the vice president, her stepdaughter went in a red jumpsuit. That was kind of interesting.
This is the J-Lo,
Yeah,
right?
that wasn't so good.
Okay.
Not my favorite. But look up Harris's stepdaughter, Emma. She came in this kind of cool red outfit.
Emma Harris?
No, I forget what her last name is, but she's Camilla's stepdaughter. She was at the ball. She looked pretty good.
Well, I need a name.
Emma.
Is it Camilla's stepdaughter?
Yeah, she's Camilla Harris's stepdaughter.
Oh, this red dress.
It's a red pantsuit, I thought.
This thing.
Yeah, she looked pretty cool.
Oh.
Thought she looked great.
She's got something in her eye.
Could be.
Okay.
Yeah.
So now, whoever asked that, there you go. You have emphasized that epi data can't be relied upon to make conclusions about transmissibility. What are the lab experiments that would?
Well, it's transmissibility in animals, right? I could do transmissibility studies in ferrets,
right?
And that would address inherent virus transmissibility. Now, transmissibility in people, those studies are not allowed.
Yep, we can't do that. Although they're doing something in England, right?
They're doing challenge studies. I don't know that that's transmissibility.
Trust science, not the scientists. Yep. But when people ask for your favorite or the scientist, that that's what you're talking about, the science that they do, right?
Yeah.
How many grants written this year, Amy?
We're on 3-4. Because 2 are due like right at the same time. So we'll have written, by the end of October, we'll have written 4.
Oh, that was Kamala Harris's stepdaughter.
Yeah, that's what I'm saying.
I didn't know it was Kamala. I just didn't know what you were talking about.
The vice president of the
I know who she is, yeah.
Her stepdaughter is the girl in the red. Her name is Emma something. I thought she looked really good for, yeah.
Yeah. Amy's working on 3 grants right now, right?
2. Well, 3, I guess, because we just had to send one. We just sent in the update.
Amy, why would obesity in itself be a risk factor?
It's an inflammatory disease.
So it exacerbates inflammatory responses?
Yes.
Hmm.
All right, one more question, and then, or what? You want me to go to 9? It's 8:55, so
Yeah.
I'll go to 9, and then I have to go plate out my cells.
How do prions fit in a tree of life?
They're not.
They're not. It's a protein, right? So they're not a living
Yeah.
thing. So they don't. I don't understand this. Are SARS spike palindromic sequences only a function of higher rates of mutation or recombination? Do you understand what the palindromic sequences are?
Well, like restriction sites, right? They're like restriction sites. That's a palindromic sequence. Like the BAM site is a palindromic sequence, right? AGGA AGG is a palindrome, right?
Okay, yeah.
So are they the function of higher mutation rates? No. Are they the function of recombination events? Probably not. They're probably just natural occurring things, like there are BAM sites in the middle of the genome, or there are eco sites in the middle of the genome. Those are palindromic sequences
they're
that are
random,
just
yeah.
natural. So no.
Do you know the current state of affairs for antivirals? Didn't Daniel recently say that he thinks there are going to be 3 by the end of the year, licensed or authorized?
Yeah.
He's got some inside information, which we don't.
It's okay.
How many changes does SARS-CoV-2 need to have before it becomes a different strain or a new virus?
It's not the number of changes, it's what the changes do. One change could be all it takes to have a new phenotype, a new property, and then it would be a new strain.
Yeah, it's not going to become a new virus. That's it. It could be one change is all it needs to have a new property. PCR versus nucleic acid amplification. What are the differences? It's the same thing.
PCR is a nucleic acid amplification test.
Yeah. Oh, Les likes Jason from Austin.
No.
Thank you, Morris, for your contribution. I learned everything. Well, not everything. You learned some virus stuff, right? Let's see. I'm looking for questions with our name in front. Here we go. Would a Pfizer booster for a spouse of an immunosuppressed individual who already got his 3rd shot be advisable to help protect the immunosuppressed person? If you already got a 3rd shot, why would you need another?
No, so like if your wife was immune compromised and she got a 3rd shot, should you get a 3rd shot not being immune compromised to help protect her? Sure.
It's not a bad idea. Yeah. Mona Lisa. My fully vaxxed Fern was exposed last Wednesday. Developed sore throat, congestion, now cough, no fever, oxygen great. Do they give monoclonals to vaccinated teens?
So I know this for a fact. Monoclonals have been authorized for EUA for 12 or above.
I would think if they're at risk, if they have any complications.
You have to be at risk. But the thing is, I don't really understand why you need to be at risk. I never understood why there was this limitation when, like at the beginning, I understood. Okay, we don't have a lot, but now we can just crank them out. They're in a biovac, so I don't understand why they're at risk. Why you still have a qualifier on them?
Is Lyme disease a virus-caused illness?
No, it's bacteria.
Bacteria. Let's see. Have there been any studies linking the R factor of the spread of the virus and the possibility of novel evolutionary fitter variants arising? Well, no. That's the problem, that there haven't been. They're just starting, and I hope that there will be some link between R and FIT. But Amy doesn't like R. She says it's a fraud, right?
It is a totally fraud. Yeah. So I sent you last week that somebody calculated the R of viruses that I've now started to become interested in, right?
Enteroviruses?
Well, I've always been interested in entros, but very specific entros, right? This was like, I forgot. I forgot what it was. But whatever. So they calculated, I think it was like Coxsackie 16, 71, and 6. And they calculated it to be like 9, 12, and 11, right? So that makes it on par with measles, right? However, let's get real here. What they calculated was the R value of the snotty little kid in daycare. And now if I take that snotty little kid and I remove it from daycare and I just have it playing by itself, the R value of Coxsackie and entero drops down to 0.1. So come on. It's a total fraud factor.
All right. How many mice does Amy work with in her lab? She has a lot of racks, doesn't she? Like one in the ABSL2. It's full.
Yes.
How many thousands would you say you are currently maintaining?
Thousands? No, not thousands.
No, you don't have more than 1,000?
No, so I don't have more than 500. I have 30 cages upstairs at 5.
It's 150.
150. And then the rack in the ABSL 2-3 that holds 70 cages at 5. So that's 350.
Okay, almost 1,000. What's the cage charge per day? 50 cents? $1?
Maybe it's like $1.12. It's very expensive. It's the most expensive real estate in the entire city of New York.
One mouse cage, folks, $1.12. It's per day, right?
Yeah, it's a per diem. It's the most expensive real estate in New York City.
And that comes with food and water ad libitum, and Amy gets to change your cage every week. Right?
Yes, yes. So when I was in Montreal, we used to joke that the locker room at the gym that I used to go to was the most expensive. That locker was the most expensive real estate in Montreal because it was like, I don't know, must have been like $250 to rent a locker a month or something or other. I don't know. It was exorbitant. And the person who used to rent the locker and would complain about it, and I'd be like, but you get the unlimited towels that you want to take a shower, and at your house, right, your wife only allows you one. He was like, well, I guess when you put it that way, it's all worth it. All right. It's 9:05 or 9:02. I have to go.
All right. Just switched from Binax to Veritor, not liking it. Any thoughts? I haven't any experience with either one. Do you, Amy?
No.
What is it that you don't like about the Veritor Animal Party?
I don't know.
All right, Amy. Thank you
all
again.
Right, I'll see you tomorrow.
Bye-bye.
Bye.
Oops, we don't want that on. Where did that? Here we go. Sorry. Okay. With one person, you can't have the close-up cropped look. Only split screen. How about that? Now, here we go. For fun discussion, do you virologists have an opinion on antiviral properties in foods? So many things have antiviral properties. In particular, many plants have antiviral properties. Many, microorganisms that you would find in soil in different parts of the world. And it's a matter of purifying them because in their native state, they're not potent enough. Antivirals need to be potent. They have to completely inhibit virus reproduction. Otherwise, you select for mutants. And this is something you would learn if you take the virology course. So I think if you like garlic, eat garlic. But, if you cook it, it's probably going to destroy some of the antiviral properties. But I don't think it's going to have anywhere near the effect of a purified component. I know many peoples who lived before the era of medicine used these. They noticed they got better from certain illnesses by eating certain things, and that's fine. But today we can purify the components if we think it's important. Have you seen any data suggesting that calcium channel blockers protect against COVID? You know, I thought I had, but I can't remember. Calcium channel blockers. Let's look for it, because we can do that quickly. Yeah, here's one study in a journal. So some have been looked at. Amlodipine besylate, it's a Nature paper. Reduced case fatality in COVID patients with hypertension. So, you know, it's hard to know what the mechanism is, right? So, yes, there is information there. Prions, we answered. I'm looking for questions with my name in front, because that way maybe we can get through a lot. Please consider making an episode that debunks the pseudoscientific claims made by all these people. So I understand that that would be useful, but let me tell you why we don't. First, I want to spend our time explaining the science to you, which I think is more productive. And I don't think many people would watch. You know, the people who like the Weinsteins, etc., would just say I'm a fraud. And frankly, I don't need that. I mean, on the Tweevo live stream, someone said I was a fraud. Not on the live stream. I was a fraud because my explanation for that virus discovery. So, you know, there's this thing where people say, oh, viruses have never been isolated. And so I explained that they were many, many times over and over. I mean, it's just a kid could explain that, right? And they said I was a fraud. But, you know, they do that just to try and discredit me and have people not listen to me. Oh, this person said he's a fraud, so I'm not going to listen to him. That's a sad thing. You know, I guess that's why I like teaching in a college. I teach, and nobody calls me a fraud. They say, I don't understand what you mean, so we can explain it and have a conversation. That's the way you learn. You don't call people a fraud. Although maybe you had professors you thought were frauds. I hope I'm not. Yeah, we might do the Bats in Laos paper Friday with Amy on. Yeah. Conspiracy theory put forward by this person. What does he stand to gain by lying? I don't know. I don't understand what any of these people have to gain by being proponents of these theories that don't make any sense. I don't get it. But they're more popular, so they're doing something. Maybe they like the popularity that comes with it. I don't know. With success of Delta and the ability to reach the unvaccinated, could this be our last wave? Well, you know, the fewer people infected, the less likelihood that new variants are going to emerge. And I mean, I don't know. I can't predict that. I think it's a possibility if you have high immunity that you suppress enough reproduction. But, you know, Mu hasn't displaced Delta in a lot of places. So you have to be careful, right? So prions are interesting. What is it here? Vanity says prions are scary. So I don't know if in your version of the course we talked about prions. Did we have a prion lecture? Because some years I cut it out because we don't have time. But I do give a lecture on prions and unusual infectious agents. And the students those years say, oh, I don't eat meat anymore as a consequence of that lecture. I think that's pretty good. Okay, people want the Laos paper, so we'll do it. Why no Lyme disease vaccine? It's not a boring question. I think all questions are good, even repeated questions. That's a good way to teach us to repeat things. Why no vaccine? So there was one and it didn't work very well. So I think it's back to the drawing board. It's not easy to make vaccines, especially for bacteria that have complicated membranes and you're not quite sure what components to include. So we were really lucky with COVID vaccines, right? I just wish more people would appreciate it, right? Are they trying to keep humans disease-free like they keep crops disease-free via RNA interference? So many people have tried to use RNA interference to control diseases of animals. The one that has really worked is hepatitis C. Hepatitis C virus requires a small RNA, a microRNA, to reproduce. And you can antagonize that microRNA with what's called an antagomere. It's the complementary microRNA that binds to it and soaks it up, and it's a good antiviral, except there are better ones now, so that's not used all that much. So, yes, they are. But, other ways are getting more attention, I think. I was in a public facility this week in New York for an entire work shift, crowded myself and one other, only ones wearing masks, 10 hours. I'm sorry. Most of the places I go, most people have masks. So at Columbia, they're required. The subway, everybody has masks. I wear it to my studio. If I go to the studio, the incubator, I wear a mask, although not everyone is wearing one on the street, that's for sure. Some people do. And where was I? I was in a restaurant where the waiters, all but one, were not wearing masks. Very interesting. But I applaud that one young lady who was wearing a mask. She realizes she's being exposed to a lot of different people in the course of her shift, so she wore a mask. It's really good. I have been pondering, this is the emergence of a new endemic coronavirus. They all started this way concept since Vincent mentioned it. Yeah, that was from Ralph Barrick, who had mentioned it. I think he was first on in February. I got it from him. I like to identify good thoughts and perpetuate them. I don't claim to invent them. As a teacher, you rarely invent things, although I have invented some things. And I like to talk about those. So the first transgenic mouse model for a viral disease we did in my lab at Columbia. It was the first ever, and now you have ACE2 transgenics, and most of the people working on them have never heard of the first transgenic, which is for polio virus. Anyway, I digress. I like to pick up the crumbs and make a cake out of them, I guess. I'm sorry, Mike. I don't know what systemic scleroderma is. So I can't really... Systemic scleroderma is systemic sclerosis, an autoimmune rheumatic disease by production and accumulation of collagen. Okay, so it's a autoimmune disease. Now, your question is, any reason to be hesitant? You have to, so certain autoimmune diseases may be worsened, right, by your body making antibodies. So, yeah, I would consult the physician and see if an exemption is reasonable. And that person, of course, may not be able to get vaccinated and will have to be monitored carefully by testing, and if they get positive, to be given monoclonals, right? Okay, let's see what this is. Why does CDC say the presence of antibodies is not a reliable indicator for immune response in COVID recover? They do say antibody levels show vaccine gives extra protection. So the problem is that we don't know what the correlate of protection is. We don't know the level of antibodies that correlate with protection in people, right? So I'll just use some numbers. Let's say one person has 50. They have a test and it says you have 50 and you have 10. They both might be protected. We just don't know because the studies haven't been done. And so that's what they mean by that. So, and that's why I object to this idea that, immunity is waning. We don't even know what it means to wane. That's what the CDC is saying, yet they say immunity is waning, so we need a booster. Okay, that's my interpretation. If I could go back in time, which vaccine would you choose? So I didn't have a choice. I went to get vaccinated, and they gave me Mrna, which I like because it has this Italian-sounding name, right, which is silly, but I liked that it was an mRNA vaccine. I thought they were cool. I was looking at the data as they came out. Phase 1, Phase 2, Phase 3. I followed it. I liked it. I was happy to get it. But, my wife got Pfizer. She went to the same, no, she got Mrna, but I think Amy got Pfizer in New York City. I think they're both very good. So I would get the same thing. I got them 4 weeks apart, right? I think that's too close. I think 6 months to 10 months would have been much better. Yeah. But at what study would you need to see to convince you that a third vaccine shot is necessary? So, as I've said, we have to look at, right now, the vaccines prevent serious disease and death. If that number changes, if it starts to go up, that's the indicator that we need a booster. I don't think it's going to go up. There's no reason for that. We've gone through Delta, and it still protects against severe disease and death. What's going to happen to make disease and death go up? So I don't think it will, but if it did, that would be the metric I would look at. Are there any downsides to taking a third dose? I don't think so, but some people say if you overboost the immune system, it could start making antibodies against your own components, right? So that could be a downside, but I think one boost is not going to do that. If you had to do it every year, maybe that would be an issue. Oh, since being on Lex, I've been addicted to your channel, so full credit to him. Well, thank you, Lex Friedman. Appreciate it. I mean, I welcomed the opportunity, which was suggested by someone here on the stream, so thank you. I will always do things to promote science and get more people to listen. And so welcome. But I have noticed that the subscription numbers on YouTube have accelerated since then. How long will it take before Pfizer for 5 to 11 will become available? Well, at least in the U.S., the idea is the end of this year or the beginning of 2022. What's up with the R1 variant that infected 45 people in a Kansas nursing home? Let's look it up. Oh, look at the headlines. Dangerously mutated COVID variant detected in 47 states. Let's see, what are the changes? That's what I would like to know. It is a new variant first found in Kentucky at a nursing facility. And what are the changes? Of course, the popular press are not going to identify the changes, and I don't want to spend any more time. I haven't heard much about it. Stay tuned. If it's a big deal, we'll talk about it. There was great discussion of observational studies and confounders at the last VRBPAC meeting. Yes, this is a real issue with observational studies. There are all kinds of confounders, and you have to appreciate that. And that's some of the issues going on with the Israeli data. Can mRNA vaccine be made for intranasal use? That's a good question. Where did I see this before? Maybe last week. So, I don't know if there are enough antigen-presenting cells in the nasal epithelium to make it an effective vaccine, right? Because the way that mRNA vaccine works is that lipid nanoparticles are taken up by antigen-presenting cells, as well as other cell types. And the protein is made that's processed and presented to T cells in the lymph tissues. So, I don't know the answer, but that would be my concern, that there are not enough sites for the nanoparticles to be taken up and be properly processed. Whereas, of course, in the muscle, it's a big tissue, and you put the vaccine deep with a long needle, 1-inch needle deep into the tissue, and you really get the nanoparticles taken up in a lot of sites. So I think that could be lacking in the nasal mucosa. Any thoughts on the prevalence of SARS-CoV-2 once everyone seroconverts? Yeah, I mean, I think, as we have said many times, it transitions to a common cold coronavirus. When that happens, we don't know. And there will be seasonal episodes, sort of like we have with influenza. It will become milder because once the population is largely immune, you prevent the serious disease, right? Then it becomes a cold. So that's probably the outcome. When it happens, I just don't know. Immune is way better than Node. Okay. I went with immune, but I thought Node was cool. It's obscure. Nobody would know what it means, and maybe they'd listen. But Node is a thing where things come together. Someone once called me a hypernode. I don't know if you know what that means, but at a meeting, a scientist came up. She said, "I'm going to stand next to you because you're a hypernode." So you guys can figure that out. I am nice to Dixon. It's just that he shouldn't have let urban agriculture fall. He wasn't willing to put in the work. Okay? And I think vertical farming is very exciting, and there's a lot to talk about it. He didn't want to do it, and I can't do it. So we'll let other people do that podcast. It's just a shame, right? Pan-corona vaccine. Oh, that's going to be a while. First, we need a pan-corona, and then we could combine it with a common cold vaccine. So we're talking a number of years, okay? A number of years. How far are we from the antiviral tablet? I think the end of the year. The only thing... I'm looking at the next question and getting distracted. The only thing I'm worried about is if we just have one, we're going to get resistance immediately, all right? Because it's going to be overused, and it then will be useless within a year. So we need to use more than one, but I'm sure that's not going to happen initially because they're being tested singly, right? I think it's going to be somewhat of a disaster, frankly. What's your favorite thing? Don't answer it. Yeah, my passwords are not based on my favorite things. Not at all. They're all the random crap that the password generators generate, not that I can't even remember, right? COVID vaccine for pregnant women. Daniel Griffin, the doctor in the house, has always said it is safe in pregnant women, and it should be given to them because they suffer from severe COVID as a result of immunosuppression during pregnancy, for sure. Is there a test for polio? Well, yes, of course. There's a PCR test that you could take someone's stool because polio virus is not a respiratory virus. Of course, it's transmitted by fecal oral contamination. It comes out in your stool and then it contaminates someone because people's hand hygiene is terrible, right? And so we could do a PCR test on stool. So is there any chance of it coming here? I would guess that it's already here because we are using an activated polio vaccine mostly, which is injected. It doesn't protect your gut from infection. So polio viruses that enter the country can establish in people's guts and circulate. But we don't look for it because that would, as someone at the CDC told me, they don't know how to explain it if they find it. They wouldn't know how to explain it to people. Oh, we have no polio, but the virus is here. So we don't look for it. And so mainly you look in places where there are outbreaks, and then you scan stool specimens for the virus. Thank you, Lynn, for your contribution. My 85-year-old father awoke Saturday with sudden worsening of dementia. Was thinking infection, but then discovered he'd had his booster the day before. Third dose, the cause. It's possible, he could have had an inflammatory reaction, which, the cytokines could have participated in exacerbating the dementia. Maybe he had a little hypoxia as a consequence. But all of that is transient, so it's good that it wore off. This is the first live stream outside of a poker live stream I have tuned into. I feel like I'm moving up. It's pretty funny. How do people find live streams anyway? I mean, I have some that I follow, right? But nobody ever pops up a live stream and says, hey, you might want to see this. Or maybe they do and I don't pay attention to it. Are furin cleavage sites naturally found in SARS viruses? Yeah, they are. They are found in a number of different, both human and animal SARS-like coronaviruses. So, I could give you a lot more details, but the answer is yes. Will you ever be doing a TWIV fungus series like This Week in Fungus? If someone has the idea, I will do it. That's the That's the only roadblock to doing new pods. So, for example, This Week in Neuroscience, Ori Lieberman contacted me a couple of years ago. He said, I want to do a neuroscience podcast. So I said, okay, I'll facilitate. And I do. I produce it for him. I participate. So if someone said, I want to do This Week in Fungus, I do it for sure. But I cannot do the legwork to find people who would want to do it. It's hard to find people who want to do it. That's why if they come up themselves, scientist who's done research on fungi, absolutely. I mean, I know some people who would be great, but I'm sure they don't want to do it because in general, people with good research programs don't want to do podcasts. Amy, of course, is an exception, and I'm grateful for that. Prions, here we go. Amy's making a painting. I don't know. Amy's gone. I'm sorry. I didn't know she was making a painting. That could be. Laos is the first nation south of Yunnan that the Lancang Mekong River runs through. A smart Twim commenter has been suggesting this for 12 months. That would be Amy. Yep. You bet. And that's why she's in part a little annoyed with the lab leak notion. It's a notion because we don't even know where the virus came from, and now it looks like it could have been even further south. Harvard and MIT have developed a small, cheap at-home test for variants using a saliva sample, isothermal nucleic acid amplification, and CRISPR. Results are visible in an hour. That's cool. I think that some of the rapid PCRs are 15 minutes, though. Also, isothermal approaches, right? Any data on non-mammalian species infected with SARS-CoV-2? I haven't heard of any. No, I'd be surprised. I mean, there are coronaviruses of non-mammalian species, but I don't know of SARS-CoV-2 infecting them. I'm sure people have looked, and the fact that we don't hear probably means it's negative, but we'll see. Get Dashak back on. You guys want to have a live stream, huh? If he has something to say that's new, I'm not sure what new. And if you just want to attack him, I'm not going to bring him on for that. What about hyperimmune immunoglobulin? We don't need it. We have monoclonals now that are highly specific and highly purified, and they work. And so I don't think we need to have immune globulin. I wasn't on Rogan. I was on Lex Friedman. I'd go on Rogan just to get more of an audience. I know people don't want me to do that, but I do it. I think it's important. The one Laos bat corona they found, can they prove it's related? They can show that it's highly related. They can't prove that it's the source. But listen to TWIV on Friday. We're going to talk about it. Now that I see that you want to hear it, now that I see that I want to hear it, we'll do it. Is there sufficient evidence to dismiss the notion that a close ancestor of SARS-CoV-2 may have circulated somewhere in humans as an innocuous pathogen evolving into a better? Well, we suspect that early on it did circulate undetected, right? And maybe didn't cause disease, but we don't have any evidence for that, no. I mean, there's some anecdotal evidence from Italy, right, that in parts of 2019 where we didn't think the virus was elsewhere, supposedly some positives, but they're not very well done tests, so I would not take them seriously at all. But perhaps evidence will be forthcoming. Where did the idea of vaccine passports come from? Look, when I was a kid, when I was 8 years old, my parents took me to Italy, and we needed a smallpox vaccine passport. It was in our passport. So the idea is not new. It's been done a long time. And studies, I'm not aware of any studies. You know, I still, do I go anywhere that require? No, I go places that I should be vaccinated, but I don't need to show it. Of course, COVID is revitalizing that. So, you know, whether you like it or not, it's going to happen. I'm sorry. Can a virus make a species jump from a reptile or amphibian to a mammal? It could, but I think it's highly unlikely because the further you get away from mammals, the cells are different, the receptors are different. So very unlikely. You know, I'm thinking of all the jumps I know of, they're from mammals to humans, right? So I don't think so. But I would never say never, right? Okay, so here's an example of what I was talking about earlier. Does a score of over 20 in the Quest Semi-Quant spike antibody test indicate neutralizing antibodies that would preclude infection? I think 20 or higher is a good score. Whether it prevents infection, we just don't know. You know, 10 might prevent, 5 might prevent. I think it's likely that it would, but we just don't know, and no one should tell you that it does. There used to be a lozenge of zinc called Ebbies that made claims of shortening common cold. Yes, zinc has been purported to, yes, shorten the length of the common cold. Actually, some trials were done. We actually worked on zinc for a while. I had a student working on it, and rhinoviruses, common cold viruses. Amy worked on it for a while, wrote a grant application on it that was not funded. Not convincing antiviral evidence, at least in cells in the laboratory. And so I don't think it's worthwhile looking at for COVID. Why doesn't a polio vaccine need a booster? It does, actually. For the oral vaccine, you get 3. You get 3 doses because, well, at least you used to, when there were 3 serotypes. Now, what do we give? Types 1 and 3 only. And so I guess you would get 2 or 3, 1 dose for each of the serotypes. And IPV is also multiple doses as well, 2 or 3. I don't remember which one. Have all smallpox samples been destroyed? No, there are a few left that we know of. In the U.S. and in Russia, and perhaps some we don't know of, and those still exist, and they're still debating whether to destroy them or not. What's a nanobody? A nanobody is a kind of antibody molecule. So antibody molecules are Y-shaped proteins, and nanobodies are not Y-shaped. Let me see if I can find a nice, it's a single domain and a single chain antibody. Let me get you a picture here to show this. Here we go. This is a good one. I like this one. Just have to click 10 times to get the picture for you. Here we go. That's a nanobody. So antibody, I guess I hit a, is that German? I'm not sure. So that's an antibody that human makes. It's a 2-chain molecule, 2 heavy chains and 2 light chains. These are all proteins, and they site where the antibody combines with antigen is up at the tip, so there are two combining sites. Then you can make heavy chain only antibodies, and then you can make nanobodies, which, as you can see, is just the combining site piece alone, right? And you can combine them. You can make 2 or 3 or 4 in a row. And these are very easy to make. You can make them in bacteria, so they're cheap, and they have therapeutic uses. Yeah, that's what an antibody is. Does that make sense? See, that's what I could do on a Virology Live. You could say, wait a minute, I don't know what an antibody is, and I will explain it. Because a lot of things I don't explain because I assume you know them. So I think a live stream can be a unique learning experience. On the other hand, it could last a long time, but I want to try and cap it at 2 hours because, well, we all have other things to do, right? How hard is it to do gain-of-function research? It's not hard. We've been doing it for 40 years. You just can make a change in a virus and see if it has a new property. Or you can passage the virus in, say, a mouse until it becomes able to grow in the mouse, and then it has a new property. It's pretty easy to do. Everybody does, many people do gain-of-function research. They give their organisms new properties. When I made mice, not me, but my graduate students in my lab, when they made mice, transgenic mice, susceptible to polio, they gave them a human gene. That's a gain of function for the mouse. So it's not just viruses and bacteria. It's gain of function for the mice. You know, if you want to make a different tasting cheese or wine or bread or beer, you could modify the yeast. You could change the enzyme, tweak it so it has a new fermentation property, and that would be a gain of function. So it really happens at every level. You know, if you and it goes beyond living things, obviously, right? You take an airplane and put a new motor on it. It's a gain of function. You can go faster or higher or whatever. Not even halfway through the questions. Look at that. Yes, NIH is testing a spike nucleoprotein vaccine. Yep. There are going to be a lot of things tested. Thank you for sending that, Lucky Sixes. For sure. Is test to stay in school a good idea? I do think so. I think in many schools where they're not allowing masking and you can't get vaccinated because they're too young right now, I think testing is a way to say, okay, this person is positive. Stay home. We'll test you again tomorrow. And if it were a false positive, you can go back. If it's positive again, we'll wait till you're negative. I think it's great. What would be your best guess as to the percent reduction in transmission from an asymptomatically or symptomatically infected vaccinated individual? Great question. No idea. There's nothing out there that's going to give you a percent reduction in transmission. We don't have enough work. That's a good study that should be done, but we don't have it right now. But very good question. That's an actionable number, right? And here we go. We can extend the nanobody question. I heard, man, I have flies buzzing around here. That's not good. What are the nanobody question? Scientists are using camels and llama nanobodies to help to develop a nasal spray to reduce load. Yes. It's not about the camels passing on Mrnas, but the camels and llamas and alpacas, they make camelid antibodies, single-chain antibodies, which you can modify just to have the binding site. So single-chain antibody would be one of those chains. You know, this is an example. Just one chain. These are two heavy chains here. Single chain camelids make those, and then you can engineer from those the nanobodies. And so really good technology. Yep. Okay, here's something I know all about because I spoke to a reporter about it today. Papers show they were hoping to introduce human-specific cleavage sites to bat coronaviruses, make it easier for the virus to enter human cells. And so this was in work proposed by EcoHealth Alliance along with people in North Carolina and the Wuhan Institute of Virology. These are documents that have been released under a Freedom of Information Act, You can read. I got them. And they were going to introduce human-specific furin cleavage sites into spikes and see what it did for cleavage, and also introduce them into pseudotyped viruses and maybe even SARS-related coronaviruses from bats. But I want to emphasize that these are not related. They're not close enough to SARS-CoV-2 to be the precursor. So it didn't happen that they did this and then it got out and it became SARS-CoV-2. Not even clear that they did the experiments. I think the grant wasn't funded if it is a grant proposal. So they were hoping to introduce it, but I'm not sure they did. But even if they were and they did, I'm not concerned. I don't think it's a risky experiment. So I did talk about what those papers meant today with someone. And I told them, not a concern. Hello, Mr. Ozzy Kim. Thank you again for your contribution. When estimating levels of virulence, should we be making the assessment in a naive population? Yes, of course. If you want to know how virulent a virus is, in humans, you can't do the experiment. You can make observations. And there's the key, right? It's going to be an observational study, so it's going to be fraught with possible confounding effects. You could do it in animals, but you wouldn't use an immune animal, right? Because then you wouldn't get infection even. So, yeah, you have to do it in an immunologically naive population. So, in humans, if you want to do an observational study, you can't do it in a highly immune population like we have. It's just not going to be, informative. I thought FDA decided on boosters today. I thought they approved boosters for over 65, right? Why doesn't the U.S. have those $5 COVID tests? It's apparently even less than $5 in Europe. It's like free in Germany. Someone wrote to us from Germany. They said it's free here. You can go pick it up on the corner of the street. I think there are a lot of reasons. I think the economics of medicine is screwed up here in the U.S. There are a lot of companies who don't want prices reduced for a variety of reasons. I think it's all economically driven. Scientifically, it can be done. As you can see in Europe, it's done. And I think it's highly subsidized in European countries as well, which is as it should be. So I don't see why we can't do that. I don't understand how medicine goes here. It's really, really quite puzzling. Would I care to send a strongly worded letter to our provincial government in Alberta on the importance of masking? I'd be happy to do that, but they're not going to listen to me. I'm just a virologist. What do I know? Why hasn't India experienced Delta as Israel? Yeah, India has experienced Delta. It's the dominant virus there, and it was the virus that made that huge outbreak of many, many millions of people some time ago. It's quelled now, of course, because population immunity went up and it's gone down. But it did rip through the population as they came out of lockdown and had big gatherings and so forth, right? So I think it did that. I'm not eligible for a booster because it's only approved for people who have received Pfizer. Yes, good point. But even if I were, I wouldn't take it. Exactly. But Amy is eligible. She's not over 65. She got Pfizer. Okay. Thank you, Ralph, for your contribution. Grateful cult member. Yeah. Are there studies comparing disease outcome in vaccinated versus unvaccinated once hospitalized? Yes, for sure. Observational studies. Right, these are not controlled studies, but we're saying let's see who comes in and what's happening to them, what their vaccination status is. And you get these periodically in the literature. You get them preprints, you get them in MMWR from the CDC, and eventually they get published. So yes, they are coming out. Thank you, Ditching, for your contribution to the incubator. You need to go on Michael Gervais podcast, Finding Mastery. Okay, that's good. I'm going on another pod. What's the name of it? Maybe some of you have heard of it. It's called Keeping It Real. Now, don't tell me it's a disaster. Okay? It sounds a little, it's one of these health lifestyle thingies. But hey, I can help out, right? I can set things straight. What else am I doing? Can't remember. Anyway, if you want me to be on a pod, suggest me. I'd be happy to do it. I'm not here for virus. I'm here because Amy is such a character. That's great. I am happy to hear that. If an asymptomatic person with ongoing replication gets an mRNA vaccine, is there interference and recombination with the next generation particles? No, not at all, because the mRNA vaccine is mRNA, which is not going to be, if it were copied by the polymerase, if it somehow got into a cell that were infected, and it were copied by the polymerase, it would just make a negative strand, which would then have to be copied again to be positive strand, and it's not the right orientation to get into to be a recombinant. So no, I don't think that's going to be an issue. And remember, it's injected into the muscle. Likelihood it's going to encounter a virus-infected epithelial cell is really low. Could you bring someone on or talk about amplifying mRNA technology? Yeah, for sure. That's an interesting tech where you have self-amplifying mRNAs. I'm not sure you need it, but it is interesting. My puppy seems somewhat lethargic following a kennel cough infection. Is long kennel cough a thing? I'm not aware of it. But, people recover differently from different infections. So I don't think I just think your dog is taking longer to recover. By the way, this reminds me, a couple of weeks ago on this stream, we were talking about long diseases, and I said there's no long polio. And someone wrote me and corrected me, an infectious disease physician, actually in New York, and said, there is post-polio. And yeah, there is post-polio. So, you get polio, you recover your limb function through therapy, and then 20, 30, 40 years later, I don't know, about a quarter of people who had polio start to lose limb function again. They become paralyzed. It's called post-polio syndrome. It is not a matter of the virus being there. The virus has been long gone. It's a matter of you lost a lot of neurons as a consequence of poliovirus. And now when you age, you lose neurons also. So the combination, you get paralyzed. So is that a long polio? I don't. It's a matter of semantics, right? You're fine for many years, and then you get an onset of paralysis. It's different from long COVID, where your symptoms never go away, right? You have all sorts of symptoms as a consequence of the first infection or the primary disease, and then those remain with you. So I think it's slightly different. But, many virus infections can have longer consequences, right? So I'm thinking of mumps virus infection. A certain fraction of kids will become sterile because the virus infects the testes. Many people who get West Nile virus disease will get some virus infecting the brain, and they will have long-term neurological issues, both cognitive and motor issues. So for many viruses, there are longer-term consequences. It's not unique to long COVID. I think it's the constellation of long symptoms is unique for long COVID and ME/CFS and so forth. But many other viruses have their own long-term issues, maybe not as a big a constellation, I guess, is the way to put it. But, it's all a nomenclature issue. I wouldn't call it long polio. I would just call it post-polio because it happens after the initial polio. So anyway, to say there are no long-term consequences of other virus infections isn't correct. I didn't mean to imply that at all, because the more you think about it, as I have just been doing, you can come up with lots of examples. Off topic for fun. Well, Amy's gone. So someone asked me this on the Reddit AMA I did 2 weeks ago. So I don't have a favorite movie, but I really like the Italian directors, right? Fellini, Visconti, etc. I really like The Bicycle Thief, Cinema Paradiso. I really like old movies where they didn't have special effects and they just worked with actors and light and action and so forth. Those are, it seems to me that those require a lot of talent. Now, I don't mean to belittle films with special effects. I think they could be cool too, but I don't know. They tend to dominate. In the older films, you just focused on the person, right? So that I like those a lot. I don't watch enough TV to have a favorite show. And I think Amy might have a favorite movie, but I don't think she watches TV only. Thank you, Cat Vaxxer. $20 for your incubator. Yeah, appreciate it. It's looking good. It's going to look even better. And, you know, by the time you're all able to visit, it's going to be amazing. The recording part will be cool. And then there's another room, which is like a board room where you have a conference table and so forth. And I am working on it myself. You know, it's, you know, Amy has been giving me advice and helping me pick this and that. But I go there on a daily basis and work. Like the other day I went to Home Depot and bought a bunch of tools that I need to do things in the space. It's kind of cool to get all this stuff. You know, I've done that in my home and so forth. But it's kind of cool to have a project like that to work on. And then I know it's going to be useful for recording, obviously. How long does a flu vaccine last for any one strain? Oh, they can last for many years, or they can be changed every year. It really depends. You know, some years none of them change, some years all of them. Yes, it's really variable. Can the mRNA vaccine, help prevent common cold? No one's checked, and I don't think they will because these are pretty benign. Nobody really gets very sick. Very few people get ill, mostly immunocompromised people. So I don't think that's happening. Will a common cold corona infection initiate a boost? I don't think so. I think there's some conserved T-cell epitopes, but I don't think so. But that'll be looked at, I'm sure, as we move into the common cold corona season, right? What first got me into virology? Well, I've told this story many times. You know, I graduated from Cornell as a biology major. I didn't know what I wanted to do. I came home. I started working as a lab tech. I read Fever, the book about Lassa fever, and that made me want to be a virologist. And there's actually a TWIV episode where I tell my story. It's buried in the archives. You can hear more about that. When avian influenza is detected on a poultry farm in the U.S., the flock is euthanized en masse in the building it was grown out in, and the carcasses are composted on site. Yeah. It's too bad, right? Thank you, IMI Canada, for your help and support. Thank you, Tom, for your support. And all of you who come here, it's really grateful. I'm grateful for your support, but I'm amazed that you come. We have 639 people and 360 likes. And we never got above 400 likes, by the way. Would that be cool? Just because you can, right? Will I be keeping a log? You know, I was thinking I should be filming what's going on, right? It could be interesting. But it's hard to film. I need to get work done, and I don't have time to film myself, and I really should. I take pictures now and then. I guess that'll be the log, yeah. Vitamin D protecting against winter infections. So there's no question that vitamin D helps make a better immune system. So I think, as Daniel Griffin had said, it doesn't hurt. I take vitamin D daily because I think it may help. I think the harm, as long as you don't exceed the dose, the harm is minimal. So I do it. And, you know, I'm a pretty critical person. But even Daniel Griffin, Tony Fauci, so I do it. Following a jab with mRNA or vector DNA or spike protein secreted or expressed at the cell surface. So the spike mRNAs encode a membrane-bound protein. So a portion of the spike will be up on the cell surface. If it's an antigen-presenting cell, It could be sampled by a T cell. Some of the spike will be intracellularly processed by proteases, chopped up into pieces and presented on the surface to T cells. And if it's a muscle cell, it could be a target of a cytotoxic T cell. But, if that happens, it does not contribute to side effects, because there are very few side effects aside from inflammatory side effects, right? Yeah, that's right. CT greater than 32 is not infectious, that's for sure. It's probably, yeah, that's correct. If this is going to be a winter disease, do the immunocompromised have to indefinitely worry? That's a very good question. If you never develop any immunity to COVID, of course, it depends on your immunocompromised status. Like, what is immunocompromised? Do you have no antibodies? Do you have no T cells? Do you have no other cells? It varies. So for example, you could have T cells but not antibodies, and then you would be protected against severe disease and death. So it really depends. Is corona a respiratory virus that causes COVID, which is a vascular disease? It's not entirely a vascular disease. COVID is the whole spiel, okay, from beginning to end. It's the upper respiratory tract illness, which happens in most people. It is the lung illness that happens in a fraction of people, depending on age, mainly. And then are the complications, the vascular problems, the neurological problems, the gastrointestinal problems, which are inflammatory-based issues. I've never seen good evidence that the virus is reproducing in endothelial cells. Endothelial cells in culture don't count. I want to see virus reproduction in blood vessels, and I haven't seen that. A few virus particles here and there do not count. And yeah, I'm fussy. I want to see the data. And if it's not there, I'm going to tell you. Who else is going to tell you that these conclusions are not consistent with the data? Did I say live virus? Yeah, I didn't mean to say live. It is blasphemy to say it's infectious virus, but I hear it so much, I guess I got contaminated. Whoa. Is it safe to vaccinate children with Cinovat, Coronavac? I don't know if they did the studies in children, below 12. You have to check that. I'm not aware often. Oh, I know what you meant. Yeah. Live. No, okay. Virology Live. I didn't say live virus. Virology Live is the course, and it's a play on my blasphemy. That's right. It's a play on my not liking saying viruses are living, but the virology is alive. Yeah. Okay, now I got it. I'm a little slow. There's no cost. It's free to you. You can take it. I just depend on people's support to set up the studio and to pay for costs. There's no cost with YouTube, of course, but the studio costs. And everyone should know by now, 11 a.m. next Wednesday, and then thereafter, Monday and Wednesday, the schedule will be published at my blog, and I will announce it on the first lecture next Wednesday, 11 a.m. Eastern, but also I'll put it on social media and so forth. Okay, we are approaching 10 p.m. I will someday keep going to answer all the questions and see who sticks around. Oh, we just passed 400 likes. Look at that. Thank you, everyone. You're so nice. I say something and you do it. I will stay, but tonight I want to stop at 10 so that I can have a 10 a.m. lecture to medical students tomorrow, by the way, which is my first lecture in front of a class since March 2020. And, I've done a lot of teaching online, but this will be the first one in a class. We have to wear masks. Everyone's vaccinated, et cetera, et cetera. So it will be interesting. It's a COVID lecture tomorrow, 10 a.m., so I have to get an early train to get in, blah, blah, blah. And I want to be rested for them, right? Original gangsters. So let's check out the super chats before I leave. Love DWR stuff and the designers you're picking. Make it wonderful. I think if I depend on Amy, which I will, if it were up to me, I'd buy cheap stuff from Amazon. It would be there the next day. And she said, you can't have cheap stuff. It has to be elegant. I was okay, fine. But many people would argue, just get what you can and put it in there. But you'll see, it's amazing. So thank you for your support. Let's see. Here we are, Ro. Thank you for your support. I really appreciate it. You wake up at 5:30 to catch you both. That's very kind of you. I appreciate it. Really stay safe there in India. Passing through a lot of great questions. I'm sorry to pass them up. But, most live streams don't get so many, I think. Well, some do, of course. Thank you, Susan, for your contribution. Really appreciate it. Many live streams I watch don't have anywhere near the number of questions. And these are all good questions, too. I appreciate them. So bring them back next week, folks. And a lot of good questions. Why is an HIV vaccine problematic? Oh, boy. We have a whole lecture on that in the virology course. Thank you, Diana, for your contribution. Can you discuss when COVID patients are non-infectious? Colleagues are concerned 20 days is not. No. 20 days is way too much. It depends what we're talking about. Are we talking about vaccinated people who are getting an infection? They decline much quicker. 10 days maximum. CT value 32 or higher, they're not infectious. There's no question. You stopped following Brett and Heather after seeing you on Lex. Well, that's cool. I'm glad I made an impression. I was really careful on Lex. They gave me a lot of time, and I answered really carefully. Do the fittest variants come from immune-compromised patients or any person with even slight disease? I don't think we know. The assumption is that they come from immune-compromised patients where there's a lot of in-host variation, right? Because the virus is reproducing for a long time. I'm not sure that's necessarily true. And I think that's going to be hard to figure out. All right. Let's see. Some more contributions here. Get through and thank them for their support, because without them, that picture I showed you, none of that could happen. Aiden, thank you. One question. Would antibodies shared in pregnancy be enough to provide sufficient protection to baby? Yeah. It's the, baby doesn't eat a lot, right? And then 6 months or so start to make their own. So, yeah, that works. That works. And yes, zinc keeps failing trials. That's right. They've done a lot of zinc trials, and they do not work for other viruses. Yeah. More. Here we go. Overjoyed, thank you so much for your contribution. Really appreciate it. And I do want to say again, you can't say it too much. Where is that? Oh, the picture is back with the Amy thing. Let me... it's gone. Sorry. Here's the Amy thing. This picture. That couldn't happen without your contributions. That's you. All that stuff is you. The desk, the sound pads, the camera. Actually, the tripod was a donation from Dixon. The computer there on the table, the switcher, the keyboard, all this other stuff, the little light, the monitor here, and more. Couldn't happen without you folks. So thank you so much for your contribution. And if you have questions that weren't answered, please don't be offended. It's just a lot, and we'll get to it. If virulence increased, thank you, Mr. Ozzy, for your contribution. This is like your fourth tonight. Could it be selected to aid transmission in the vaccinated by inducing symptomatic disease and potentially making it more serious? If virulence increased, could it be selected to aid transmission in the vaccinated by inducing symptomatic disease? I don't know what the... So you're saying that an increase in virulence would help transmission. I do think that is one of the reasonable explanations for an increase in virulence in a virus, evolutionary increase, where it would facilitate transmission. Because if it does not, what would be the selection for it? Right? I don't think there would be any unless it facilitates transmission. So I think it's theoretically possible, yes. John, thank you for your contribution. Yeah, I saw this today that Mrna leaves antibodies higher at 6 months. No, memory and T cells are formed earlier. I'm not sure that, well, I saw the announcement. I have to look at the paper before I say further. Let's take a look and see how much it is and what the ideas are. Yeah. There's great comments here, folks. Really great. Sorry, I have to skip over them. And I'm just looking for the final. I think that's it for the super chat. We got 485 likes. Now we have 600 people here. What else do I need for the incubator? So I need sound curtains for the windows because it's too noisy. I need sound curtains for the door behind me. I need a couple of more lights. I need another camera for the interview. The furniture is ordered. It's coming in. And then I have a monitor stand to put that monitor that was on the box. And I have another desk coming in. So that desk you see there is the work desk where the editing is going to happen. That's not going to be where the filming and streaming happens, but it's temporarily being used for that. And when the new desk comes in, I'll be recording there, and then the work desk will be moved. The work desk will have an editing station, a computer, which I already have, and a monitor, which I have to get. So not a lot more needs to come in. I've spent quite a bit of money there, and we're almost done. So I should be completely tricked out by the end of October, I think. All right, folks, thanks again for coming here. Really appreciate your being here and asking great questions. Stay safe, and come back next week. Good night.